My name's Todd Lane. I'm the Deputy Dean of the Faculty of Science here at the University of Melbourne. I begin by acknowledging the traditional owners of the unceded lands on which we are meeting here tonight, the Wurundjeri Woi-wurrung people. I acknowledge the traditional owners of the unceded lands from which you are joining from as well. I pay my respects to elders, past and present, and I welcome any First Nations people who are joining us this evening. It is my great pleasure that I welcome you to the final of the Big Science Pitch for 2025. Our Dean, Professor Moira O'Brien, is currently overseas and sends her apologies and her congratulations to the finalists. The Big Science Pitch showcases some of our amazing research from across the Faculty of Science, and we deeply appreciate you joining us here tonight. The competition originated in 2021 in the School of Biosciences, and last year we expanded it to our entire faculty, opening applications to all 7 schools. This year we have 7 finalists selected from a record number of applicants. They come from 4 different schools. 3 from the Melbourne Veterinary School, well done MVS, 2 from the School of Physics, 1 from the School of Chemistry, and 1 from the School of Geography, Earth and Atmospheric Sciences. Each of our finalists have already won $10,000 by reaching this stage of the competition. But tonight, they're competing for an additional prize pool of $40,000 in research funding. $15,000 for first, $10,000 for second place, and $5,000 for third place. This is decided by our panel of judges, who you will meet soon. I'm one of them. There's also a People's Choice Award that you get to vote on, and that's for $10,000. Each of our finalists will present a 3-minute pitch for their research idea, and in this short time, they need to persuade the panel and the judges and our public audience that their proposal has the highest scientific innovation and potential societal, economic, or environmental impact. Knowing the level of talent in this group and the quality of presentations, this is going to be tough. The Big Science Pitch shines a spotlight on some of our remarkable early career academics. We're passionate in the Faculty of Science about supporting our staff and especially keen to support our early career academics as they grow in their careers and become our future leaders. The Big Science Pitch is one way we're doing this, providing support for our emerging leaders to hone their skills, pitching their ideas and communicating to a general audience. I hope that all of our 2025 entrants, including those who didn't make it to the finals, have found this competition a useful experience. Well done to all of our competitors for having the bravery to submit your pitches. And now, I'm going to introduce our MC for this evening, Dr. Lyndon Ashcroft. Lyndon is a senior lecturer, a climate researcher and science communicator in the Faculty of Science. Lyndon's research explores past climates of Australia, with particular focus on using historical documents like newspaper articles and weather observations to understand past extreme events. This is critically important for also understanding our future climate change. Lyndon's research, teaching and outreach has been recognised with several awards, including being a Science and Technology Australia Superstar of STEM, a Victorian Tall Poppy, and receiving the Australian Meteorological and Oceanographic Society's Outreach Award. Lyndon is often interviewed by the media, and she loves talking to schools and community groups about science communication, climate change, and climate action. We are in safe hands with Lyndon, so I'll hand over to her, and she can take the lead from here. Thank you, and enjoy the evening. Thank you.
Thanks, Todd. You do have a tough job this evening, and I have a wonderful job. Welcome, everybody, to the Big Science Pitch for 2025, possibly the most exciting way you could be spending a Tuesday evening or a Tuesday morning, depending on where you're joining us from. As Todd says, this is our 5th Big Science Pitch, but only the 2nd pitch that's been open to the entire Faculty of Science. I know many of you are joining us for the 4th or 5th time, so welcome back. And those of you who are joining us for the first time, we have a record number of people watching this evening. We're really glad that you're here. I would like to know, as we get started, where you are joining us from. So please, in the Q&A section, let us know where in the world you're coming from, and I'll do some shout outs in just a minute. But while the hellos come in, I wanted to reiterate what Todd said. We have a very esteemed judging panel here tonight. You'll get to meet them in just a little bit, and they have a really hard job picking first, second, and third from our 7 finalists this evening. But you also have a hard job because you will be helping us award the People's Choice Award, that extra $10,000 to help these researchers progress their research to help change the world. You are in charge of deciding this one, and you will get to cast your vote once all 7 pitchers have presented their research. Oh my goodness, we've got so many people coming in. There are people coming from Melbourne. Someone's calling in from Ballarat. Someone's calling in from Hong Kong. Hello, it's so wonderful to have you with us. Werribee, the Gold Coast, all of these amazing exotic places. Newcastle as well. We've got people dialing in from all over the place. A special bonus if anybody wants to guess maybe the youngest and oldest viewer possibly watching, if we've got anybody in the 90s or anybody in the sub 1-year-old age bracket, that would be interesting to know as well. People are here from Clarinda. Somebody's here watching from Vancouver, from Adelaide. How lucky we are to have you all here with us this evening. Many thanks for joining us. All right, Hobart as well. They keep coming in. It's really great to have you. But we are also on a tight schedule and I don't want to keep you on the couch for too long, so it's time to get started with our first pitcher. Our first pitcher this evening is Dr. Gayatri Gopakumar. Gaia studies host virus interactions and develops vaccines for animal viral diseases. She holds a master's degree in biotechnology and a PhD in virology from the University of Melbourne. Gaia has led clinical trials for the first deletion mutant vaccine for infectious laryngotracheitis, I practiced that one, in poultry, developed by her lab group and secured its registration in Australia. Her current research is focused on developing a vaccine for the herpes virus that affects marsupials. Over to you, Gaia.
What makes Australia truly special? Well, I guess you have a lot of answers to this, but I think it is our marsupials, our kangaroos, wallabies, and the quokkas that smile in selfies. Unfortunately, these marsupials face a lot of threats, and we all know about the obvious ones: climate change, bushfire, and habitat loss. But there is an invisible threat, one that most people haven't heard of: marsupial herpes viruses. You can think of these viruses like the weeds in your garden. They may dry out at times, but the roots stay hidden and they grow back, sometimes even stronger. These viruses cause respiratory to neurological diseases, sudden deaths, and even affect the ability of these animals to reproduce. And the scary thing is that all of Australia's marsupials are vulnerable to this infection. Even worse, the very places designed to protect them, such as the captive breeding programs and sanctuaries, can become hotspot for the transmission of these viruses to new host species, causing disease outbreaks. And it doesn't end there. Once these captive animals are released back into the wild, they can spread these viruses to the uninfected wild populations. These events not only affect the animal health, but also compromises the wildlife conservation efforts and the resources invested in protecting the critically endangered or threatened marsupial species. So, what's the solution? Well, we can think of 2. 1, separation, like the lockdown we humans experienced during the COVID crisis. But this is not practical. These animals need a normal life. They need to hang out together, to breed and bond and just be marsupials. So that leaves us with the second and the only realistic option, which is vaccination. So why haven't we done that yet? Because no vaccines exist until now. With my previous experience in vaccine development work, I'm thrilled to tell you all that I am developing the first ever herpes virus vaccine for marsupials. This is an inactivated vaccine, and so there is no risk for viral spread, and it is safe for even the vulnerable marsupial species. By developing this methodology, I will also make a simple and practical system for producing custom-made vaccines directly on-site, right where they're needed. I'm also excited to say that infection studies for this project are already underway using an animal model. With your support, I can take this vaccine from lab to landscape to protect not just individual marsupials, but to reinforce the whole wildlife conservation efforts that safeguards these animals. Thanks to my research, I'm hopeful that long into the future, we will still be able to get that selfie with a healthy, smiling quokka.
Thanks, Gaia. I really would love to get a picture with a healthy, smiling quokka. It's really great to hear about your research. One thing I forgot to tell everybody watching at home is that the other thing I need you to do, apart from pay attention for the People's Choice Award, is to ask some questions. If you can think of a question for any of our pictures, pop it in that question and answer box as soon as you can, because they are being beamed directly to this iPad that I have here, which means I get to ask them. And I have a few questions that have come in while you were doing your pitch. Question one here from Emily about how you realistically deploy a vaccine. Like, what are the barriers to deploying a vaccine like the one that you're developing in the wild?
Yes, so that's a very good question, especially because our targets are wild animals. So currently the vaccine that we are developing will be targeted for animals, marsupials that are going to come under captive care. So the virus that we are developing is for marsupial herpes viruses. So essentially what we have seen is that herpes viruses spread when animals come in close contact with each other, which is why animals, when they are brought to captive facilities for breeding and for other caring purposes, that is where there is the highest chance of transmission of these viruses happening. So our initial plan is to vaccinate these animals when they come for captive care and then release them back into the wild once they are vaccinated.
Right, that makes sense.
Yeah.
Okay, then we have a few questions around the human component of this, as you can imagine. Kedu wants to know whether humans can get infected with this type of virus and whether you think these vaccines could then be expanded to humans eventually. That's from Amy.
Okay, so coming to the first question, which is, can marsupial herpes virus infect humans? Based on our scientific reports, so far there is no evidence to say that marsupial herpes viruses can infect humans, or any herpes viruses of a non-marsupial can infect marsupial species. So this is because herpes viruses have co-evolved with the host for millions of years, that they have become adapted to living with one single host species. Now, having said that, we know that marsupial herpes viruses can jump from one host species to another host species. Like we have seen cases where herpes viruses jump from wallabies to eastern grey kangaroos, and from eastern grey kangaroos to water opossums and Virginia opossums in a captive facility in the United States, which actually led to the death of all of these animals. So yeah, that's my answer for the first question. And the second question, if I may ask again?
Was whether these vaccines could be expanded to humans eventually, but it sounds as though we don't need that because these particular...
So each, usually the herpes viruses are very much adapted to a single host. So if we have to develop a vaccine, for humans, it has to be like a separate vaccine. Yeah.
I get that. Okay, we've got time for one last quick question, and somebody's asking whether what the long-term impact of this vaccine could be on genetic health and survival of threatened marsupial species.
Yes. So the long-term
impact of this vaccine. You know, what could you imagine the long-term positive or possibly negative impact could be if once this vaccine is hopefully rolled out?
Yes, so that is a very important question. So during my pitch, I said what damage the herpes virus can cause during an infection to the marsupial species. But what we also need to understand is that this is not like a standalone event. So there are certain marsupial species where herpes viruses infection are often associated with infection with other pathogens. And when that happens, like a co-infection, the outcome of the disease often becomes more serious and complicated than infection of the original animal. And apart from that, when we look at, the clinical signs of a herpes virus infection of marsupials, it may not sound or seem as dramatic as infection with certain other pathogens of marsupials. But that is kind of the, modus of operandi of herpes viruses, because once herpes virus infect an animal, the infection is going to last lifelong in that animal. And we already know that a lot of Australian marsupials are already exposed to these herpes viruses. And with more and more problems that we are facing with climate change and habitat loss, we are expecting more and more of our marsupial species to be listed as endangered or coming into the critically endangered or threatened category, which essentially means that more and more of these marsupials are going to come into captive care, which is where herpes virus outbreaks often happens. So this is why herpes virus infection of marsupials are considered as a quiet but an emerging conservation threat, and why vaccination of these animals are quite essential to protect them, or a proactive measure to protect them from these diseases, and also to ensure that our conservation efforts for marsupials are going to work very well in the future.
Thank you. Thanks so much, Gaia.
Thank you.
We're going to move on now to our second pitcher, Dr. Jason Zhang. Jason completed his PhD in Systems Neuroscience in 2021 here at the University of Melbourne. He is a research fellow in physics and a former Lions Vision research fellow. This latter role recognises him as an emerging leader in medical research. His work advances high-resolution neural implants, brain-machine interfaces, and implantable systems for seizure prediction and neurological monitoring. Welcome, Jason.
Close your eyes for a moment and imagine losing control of your body. Your muscles stiffen, your awareness slips away, and when it's over, you can't remember what just happened. That's what a seizure feels like. For 50 million people around the world living with epilepsy, the fear that a seizure can strike any time without warning is constant. It's not just dangerous. The unpredictability of a seizure changes how you live your life. You cannot drive, pick up your kids, or even hold your baby. I study the brain because I believe we can change that. Most treatments for epilepsy rely on tracking seizures, which right now is like trying to predict a storm by listening for thunder. By the time you hear it, the lightning has already struck, and by then it's too late. But what if we could see the storm forming and step in before it breaks? In physics, there's a concept called criticality, how systems balance on the edge between calm and chaos. The brain does this too. Most of the time it's stable, but sometimes activity builds and spreads until the brain suddenly tips into chaos, and that's when a seizure begins. My research shows we can spot signs of getting close to that tipping point before the seizure starts, like seeing the first grey clouds on the horizon. Our next challenge, however, is to find that signal fast enough. Right now, we record brain activity, send it to powerful computers, and spend hours analysing it. It's like collecting weather readings and waiting until tomorrow to find out if it rained today. To stop seizures, we need to act before they happen, not hours later. We need a system that senses early changes and makes decisions very quickly. A built-in weather forecaster for the brain. That's what my project is building. I'm developing algorithms that learn when the brain is getting too close to the tipping point. We're designing smart software that drives a tiny implantable chip to record the signals in the brain and give warning when a seizure is about to strike. Think of it as turning a bulky weather station into a smartwatch for the brain. And this isn't just about technology, it's about freedom. With your support, we can make seizures foreseeable, and hopefully even preventable. At the very least, we hope to give people enough warning to prepare and stay safe when the storm hits. Help us create a world where people with epilepsy can drive again, pick up their children, hold their loved ones, and live without fear. Thank you.
Oh, Jason, that photo. Thanks so much for sharing that and your pitch. It's really valuable research. We've got a lot of questions that have come through so far. Beck has the first question here, and she wants to know how close we are to seeing this real-time seizure detection technology used in trials.
So I've been working on this project for over 5-6 years. So we are at the end of the preclinical studies that we've been doing to develop this high-resolution device. And not long ago, we got our ethics approval at the Royal Melbourne Hospital to try the first-in-human implants.
Congratulations, it's great.
Thank you. Yeah, so we're working with neurosurgeons, clinicians, engineers to get this going. So hopefully next year sometime we'll have some good news that we'll have our first humans implanted.
Amazing. So another question that follows on from that is about the challenges that you think still remain. If you've got the first human trial coming soon, what barriers remain after that before this technology can be widely adopted?
Yes. So there are several challenges. So one of the issues that we have kind of seen before with preclinical study is the longevity of these devices, and we've specially fabricated these devices so that it's almost invisible to the brain tissue. And another thing that we have worked on is the chip. So instead of having a tether or a long line that goes out of the skull, we're thinking of making this into a wireless system, so that, like I mentioned in the pitch, the idea is to have it in a smartwatch or in our phones, so that patients or the clinicians can have a real-time view of the changes in the brain signals that we see with people with epilepsy.
All right, I've got one last quick question before we need to move on about the high resolution nature of the implants. Why is that so important for detecting these tipping points in seizures?
So currently in hospitals we use EEG recordings, and they are these caps that we wear, and they're basically measuring millions of neurons because it's actually on the skin. So it's actually quite far from the neurons that we want to record from. Whereas what we're trying to do is we're trying to get as closest to the brain and the neuron. So we've made these tiny implants so that we can actually listen to how the neurons talk to each other. So we're getting closer and closer to the neurons, and then seeing how they communicate. So that's the difference.
Yeah, wow, amazing. Thank you. What a start to the night. Thank you, Guy and Jason, for kicking off with such a big bang. And thank you to all of you for asking such thoughtful questions. I'm sorry that I'm not able to get to all of them. We're running a tight ship. And now I'd like to introduce you to 4 passengers on the ship who have the hardest job of all tonight, I would say. You've already met Professor Todd Lane, who is the Deputy Dean of the Faculty of Science. He's also a Professor of Atmospheric Science, but Jason promises me that he didn't use that weather analogy just to get in the good books of the head judge of the panel. And Professor Lane is joined by 3 esteemed judges here. We have Rebecca Webb from the Melbourne Veterinary School. Now, Becca's probably our most empathetic judge because she competed in the Big Science Pitch last year and she won it, even though she was in New York at the time and dialed in the middle of the night. It's great to have you in the studio tonight, Rebecca. Next to Beck, we have Imogen Crump, who is the editor of Pursuit. Pursuit's the University of Melbourne's research news platform. It publishes articles that are accessible to non-specialist, curious readers. And the website's visited by more than 1 million people every year. It's great to have you, Imogen. And next to Imogen, someone else who might have appreciated the weather analogy is the wonderful Nate Byrne. Nate is a Bureau of Meteorology trained meteorologist, but you probably know him better as the weather presenter from ABC News Breakfast, or if you are under 6, from his stints on Play School. It's great to have you all here. You have a really tough job ahead, so thanks for heading up the panel, Todd, and good luck for the decisions that you have coming up. I would not want to be in your shoes. Let's move on now to our third pitcher, Dr. Catherine Morgan. Catherine is a postdoctoral fellow at the School of Chemistry. Her research focuses on designing new personalised treatments for cancer using metallic radionuclides. She's developing molecular cages that are purpose-built to trap these radioactive metals and attach them to tumour-targeting antibodies and peptides. Thanks, Catherine.
When you think of medicine, it usually comes in the form of pills or injections. The treatment I'm designing begins with a single atom. One radioactive atom trapped inside a chemical cage has enough energy to destroy a cancer cell, if we can create the right way to deliver it. That challenge is where my research begins. Breast cancer is one of the most treatable cancers when it's found early, but once it spreads to other parts of the body, known as metastatic cancer, it becomes much harder to control. These tumours often spread to places like the bones, where they hide and grow. And over time, metastatic cancer stops responding to the treatments that we rely on. My research is developing a new way to fight metastatic breast cancer. If you imagine a cancer cell, you would see it has tiny proteins all over its surface. Think of them like molecular locks, just waiting for a matching key. Well, we found a key that fits one of these locks, and it's a small peptide with the fabulous name of bombesin. Understanding this lock and key system has allowed me to design a radioactive treatment that hones in on cancer with cellular precision. Now, if you were thinking radiotherapy is already an important part of breast cancer treatment, you'd be absolutely correct. Traditional radiotherapy uses powerful beams of radiation from outside the body, which can sometimes damage healthy organs like the heart and lungs. Often, once cancer has spread to multiple places, conventional beam radiotherapy cannot be used to treat every individual metastasis. Instead, I'm developing a treatment that can move through the bloodstream, seek out metastatic cancer cells, and deliver radiation exactly where it's needed. Picture it as a homing missile designed to find and bind breast cancer cells. At its core is a chemical cage that traps a single atom of radioactive terbium-161 as it travels through the body. Attached to this cage is the peptide bombesin, the guidance system, which steers the missile directly to cancer cells, delivering a radioactive payload powerful enough to destroy a tumour while sparing the heart and lungs from damage. In this project, we're turning a weakness in cancer biology into a therapeutic weapon using the power of precise radiation. With your support, I will develop new radioactive delivery systems and test them in breast cancer models. My research is an essential first step towards finding a new way to fight metastatic breast cancer. Thank you for listening.
Thanks, Catherine. Questions coming in, and you may guess what the questions are generally focused on. There's a few curly ones, but a few people like James and Sarah are asking about the hurdles that are in the way to get this therapy into clinical trials and how far this approach is from being translated into actually helping patients.
Yeah, absolutely. So it's a long process from, they call it from benchtop to bedside, can take up to a decade. And it involves countless partners, whether that be researchers and then clinicians and eventually industry partnership. And it's really a very interdisciplinary and collaborative process. In terms of regulations, it's of the utmost importance that we do all of our research using, ethics approval. Even at the early preclinical stage, using ethics is incredibly important. So we have to apply for ethics for any research that we do preclinically. In terms of further down the line, again, a lot more ethics. But yeah, that would be more on the clinician side.
Definitely. But I can understand why people are asking those kinds of questions. Sneha has another question. Fantastic work, they say, and I would agree. Is there any prospect of applying your research to other kinds of metastatic cancer?
Yeah, absolutely. And so I spoke about Bombesin before, the guidance system. And so this peptide binds to a protein that we call the gastrin-releasing peptide receptor. And so this protein that's on the outside of cancer cells, and in this project I'm talking about metastatic breast cancer, it's actually present on the outside of other cells, and notably prostate cancer cells. So there are different treatments for prostate cancer, but sometimes they don't work. And so really it could be applied to metastatic prostate cancer when potentially the proteins that are currently tried to be targeted, they're not responding to those treatments. So absolutely, that's a great question.
Cool. Okay, last one. About the excess radioactive molecules. You talked about targeting and how that's kind of a better approach than chemotherapy. But if there are excess radioactive molecules, this question's from Vivian, what happens to those in the body?
Yeah, okay, so these are great questions. So it's, again, we call it targeted radiotherapy because we're really hoping that the years of, synthetic chemistry, the peptide synthesis, the inorganic chemistry means that we have a really specific and targeted molecule. That being said, once it's in the bloodstream, it has to compete with every other protein, every other enzyme, and every other organ. And so what we're hoping for is that we've found this perfect balance where we get rapid uptake to the tumors or to the tumors that we're interested in targeting. We also get rapid excretion. And what that means is, for peptides and small molecules, they're often excreted through the kidneys. And so that's another layer of complexity that we need to factor in, is that we need to be thinking about potential kidney protection strategies on top of that. But really, in terms of a peptide, they can be excreted rather quickly. So often within, a couple of hours, a lot of it has been excreted. And so, yeah, that's typically where it goes.
Amazing. Thank you. Thank
No
you so
worries.
much, Catherine. And for all of your questions. Again, there's too many fascinating ones and we don't have time to go to all of them, unfortunately. We have to move on to our next picture in a minute. But first I wanted to share a bit of a story from Rebecca Webb, Dr. Rebecca Webb, who you met earlier. She's one of our judges this evening and she won the Big Science Pitch last year. And I thought it would be nice to hear what she did, basically, with the money, right? Because that's kind of what we're here for tonight. The funding that she received last year gave her a head start on building a customisable cell infection model to study chytric fungus, which is a devastating fungus that's causing death to lots of frogs all over the world. She was able to use that model to trial novel conservation strategies for the beautiful corroboree frog. Rebecca shared some of her results with researchers from other universities and conservation programs, and importantly also with traditional owners at the lands where the corroboree frogs were once widespread. So I think it's really important to hear the power of the Big Science Pitch to make research take that one step further in changing the world and making it a better place. All right, enough from me. Who wants to hear another pitch? Oh, I can hear you through the screen, so many of you. Oh, all right, let's go. The next science pitch is from Dr. Kimberly Reid. Now, Kimberly's PhD at the University of Melbourne focused on atmospheric rivers. These are streams of moisture in the atmosphere that cause really heavy rainfall. After her PhD, Kim moved over to Monash Uni, where she completed a postdoc and investigated the 2022 devastating Eastern Australian floods. She gave evidence about that event at related parliamentary inquiries. A big job for a researcher. Now at the Centre of Excellence for Weather of the 21st Century, Kim is researching hazards and resources associated with fog. Take it away, Kimberly.
Have you ever driven through thick fog and panicked because you forgot how to turn your fog lights on? or sat frustrated at the airport gate 40 minutes after your plane was scheduled to depart due to fog. You probably think of fog as a problem, but some of the driest places in the world rely on fog as a source of drinking water. Fog harvesting is used in arid and remote places globally, such as South Africa, California, Chile, and Spain, to extract drinkable water from the air without the need for rain. As the driest inhabited continent in the world, water availability is a big challenge for Australia, especially in remote Indigenous communities. Where rainfall is scarce, these communities typically rely on groundwater, which is prone to contamination. Between 2018 and 2019, at least 25,000 people in remote communities accessed water that did not meet basic health guidelines. But fog doesn't occur everywhere, so how do we know where we could actually harvest a reliable amount of water from fog? This is where I come in. My plan is to identify locations around Australia that would be ideal for fog harvesting. I know what you're thinking. How on earth do you harvest fog? A fog harvester is basically a mesh between two poles. As the moist air moves through the net, it accumulates into water droplets that drain into a pipe at the bottom. And since it comes straight from the sky, fog water is exceptionally clean. While drinking water is the main use of fog harvesting, in Spain, they also use it to help regenerate forests after bushfires. Researchers have even used fog meshes to collect the steam that comes out of power plant cooling towers and data centers. Finally, a valuable use of AI summaries. Fog harvesting is cheap and relies only on gravity, so no electricity is required. This means no ongoing running costs and maintenance costs are small. But we can't just stick fog harvesters anywhere and hope for the best. We first need to ensure the typical weather conditions at a location are suitable for harvesting fog. Using the funds from the Big Science Pitch, I will buy smaller pilot fog harvesters and place them at existing air quality monitoring sites across the university, such as the top of the Redmond Barry Building, Burnley and Dookie campuses. These sites include other instruments to measure the temperature, wind speed, humidity, and rainfall. This means I would have records of the weather and how much fog water is collected at the same location. Now, we're probably not going to supply Melbourne's water needs with fog collected from the Redmond Barry Building. But the data gathered over the course of a year will allow me to construct a model that uses weather station data to estimate how much clean fog water we could potentially harvest at different locations around Australia. This will allow me to create a map of potential fog harvesting sites across Australia, which I would share with state governments and water suppliers to help them bridge the gap in access to clean drinking water across Australia.
Hello, Kim.
Hey, Lyndon.
That's very exciting. Nice to have another weather weenie up here with us. You've got quite a few questions, but I'm going to ask the first one because I've got the iPad. About the atmospheric conditions. What kind of atmospheric conditions do you need for fog and therefore fog harvesting? And how predictable are they? I'm thinking of airports. I'm thinking of Canberra.
Well, you're right. Canberra is the foggiest airport. But for fog harvesting, we actually want the fog that's come off the ocean. The reason being is that the water droplets in the fog are bigger and get stuck to the mesh more. So to get those kind of conditions, we want coastal areas that have cooler ocean temperatures along the coast. Think sort of around Adelaide, the Great Australian Bight. We also want onshore winds and cool overnight temperatures to help that water vapor in the sky condense into a liquid. So we want moisture, cold air temperatures, and winds that are blowing onshore are the key ingredients for fog harvesting.
Okay then. Well, another question that's come through is about the biggest barriers to using fog harvesting in remote Australian communities. And possibly you've just answered that. Can you think of any other barriers there?
Yeah, so one of the biggest challenges is just having good observations to tell whether we could even harvest fog. And unfortunately, the current observations we have are quite scarce in the areas that we want to look at, sort of remote areas, particularly Western Australia. And so we really have to literally stick a fog harvester there, a smaller one, just to test whether we can even gather enough water before we start building larger arrays of fog harvesting. So there is a bit of a lead time where we have to do a bit of testing at the site before we can build an actual fog harvester, and that might be a year or so, plus a lot of community engagement. So it is a long-term project, but it could be really valuable.
Interesting. Okay, one last question, one you could probably have predicted. Climate change. Can you say anything about the impact of climate change making fog harvesting more or less likely in the future?
Well, that's one of the questions that we want to answer, so I don't have the answer, but I can hypothesize that as the atmosphere warms, it's able to hold more atmospheric moisture. So the lower atmosphere should hopefully have a bit more moisture that we can then potentially harvest. However, fog really depends on local factors, not just the global air temperature, but things like what's over the land. Is it vegetation or cement? These sort of local factors that are a lot harder to predict. And so we really have to study individual sites at that local scale. And that's what makes it difficult to anticipate how climate change could affect fog harvesting.
Interesting. Thank you. Thanks so much, Kim. Now, a couple of reminders before we hear from our next pitcher. Please make sure you're thinking about who you're going to vote for in the People's Choice Award. The voting lines will open at the end of our final pitcher. And also keep adding your questions in for your pitches. I'm getting too many to answer, but I'm doing my very best. As soon as you think of them, please pop them into the Q&A and they'll get sent through to me. Now, we are up to pitcher number 5, and our 5th pitcher is Dr. Megan Lucas. Megan is an award-winning lecturer and animal welfare scientist at the Melbourne Veterinary School. With broad expertise in animal welfare across species, she focuses on pig welfare and behaviour, studying factors such as the human-animal relationship, early life experiences, the physical environment, and, as you'll hear in a minute, thermal conditions. She's passionate about applied research that can be directly translated on farm to improve the lives of pigs and the people who work with them. Thanks, Megan.
We all want the animals we eat to have had a good life before they end up on our plate. But what if up to 40% of those animals never make it to our plate at all because they're dying well before they get there? This is the case for the pork industry. Anywhere from 5 to 40% of piglets born can die within the first few days of life. Why? Well, at birth, piglets can weigh less than a kilogram, and by comparison, their mother can weigh 300. And this disproportionate size difference leads to the biggest cause of piglet deaths in the industry, where the mother accidentally crushes her piglets, suffocating them. It's a huge animal welfare concern, a productivity issue for pig farmers, and increasing public scrutiny makes this a critical threat to the sustainability of the industry. Now, crushing occurs in all types of pig farming systems, but sometimes it can be a bigger problem in free-range outdoor systems where mum's movement is not restricted. And my research has identified a clear link between low temperatures and increased crushing risk. In cold conditions, piglets use most of their very limited energy reserves to stay warm. Little energy is left for movement, so they remain very still and rest close to their mother for heat. But being immobile and in this location increases their risk of being crushed. In outdoor systems, the pregnant mother lives in a paddock and she gives birth in a simple shelter that's called a farrowing hut. This is a partially open shelter with straw on the ground and no internal features. I want to redesign these huts to make them safer and warmer for piglets. I'll do this by adding practical features such as lightweight guardrails to protect piglets from crushing, insulated flooring, and low energy heat sources that keep piglets warm without overheating mum. I'm working in partnership with the pig industry because we share the same goal: healthy, happy piglets. And it's essential that new design features are tested carefully on farms to ensure they work in the real world. I'll also collaborate with engineers to develop effective solar-powered heat sources that are safe for animals and feasible to use in paddocks without a power source. Through practical and cost-effective changes to outdoor huts, my research will enhance pig welfare, boost farm productivity, and strengthen community trust in the industry by creating a warmer environment with less crushing risk. Rather than reinventing the system, my research focuses on retrofitting existing huts. This keeps costs low and, put simply, is the only approach that makes real, large-scale improvement in animal welfare achievable across farms right now. So if you want to be part of positive, real change in animal welfare, support my simple, hot solution to this cold problem.
Piglets. Come on. Too cute. Too cute. Thank you, Megan. It's lovely to have you here. All right, a question from Joe. How receptive has the pig industry been so far to these implementations that you're suggesting?
Great question. Extremely receptive. I think the pig industry is a fantastic example of an agricultural animal production industry in Australia that is really invested in animal welfare. In Australia, our pig industry has made a lot of voluntary moves on their own without being forced by policy or retailers to actually make improvements in animal welfare that are sort of self-initiated. And I guess from a research perspective, they're also really keen on investing in research in animal welfare too. And in terms of farmers, going down to that level, farmers are also really receptive to changes. I think more than anyone, they want good welfare for their animals.
Yeah, that makes sense. Another question about improving the welfare of early life, not just the survival of the piglets, but if you can improve their welfare in early life, what benefits does that have for long-term quality of life?
Oh, what a fantastic question. So part of my PhD was actually looking at how early experiences shape the lifelong welfare of animals. And we certainly do know that the early experiences they have with people, so with farmers, and also the early experiences that they have in their physical environment and whether they've got opportunities to engage in really highly motivated behaviours that are important for their welfare, those types of experiences early in life definitely have a long-term impact on their welfare and how well they cope with day-to-day life. And so if we're talking about early survival, that is important, but I think it's great that people are acknowledging there's other aspects to welfare as well.
Yeah. Oh my goodness. Too many great questions coming through. Do you think your research has the capacity, this is from Sage, to improve increasing consumer expectations for improved animal welfare?
Yeah, absolutely. I think anything we do in terms of improving animal welfare, animal welfare is something that's intrinsically linked to consumer trust. So I certainly think that this is something that would affect consumers' perceptions of the industry.
Okay. I'm going to sneak in one more. I think I've just got a really quick answer, if you can. The behavioural cues in piglets. What behavioural cues in piglets have been the most informative for you in designing these interventions?
Well, I guess it depends on our research question. If we're talking specifically about thermal stress during winter and cold conditions, then we're looking at thermoregulatory behaviours. And don't overthink it here, shivering, huddling in one group and resting right close to mum as well. That's something that we've identified as a risk factor for mortality because when piglets are lying right next to mum, obviously using her as their heat source, we know that this increases their risk of dying, unfortunately.
Okay. Thank you. Many thanks, Megan. We're going to stay in the Melbourne Veterinary School now because it's time for me to welcome our sixth pitcher, Dr. Mia Cobb. As the Chaser Innovation Research Fellow in Canine Welfare Science, Mia aims to improve the welfare of dogs, from working colleagues to couch companions. Big hello to any dogs watching right now. Her interdisciplinary research focuses on understanding how people think about dogs and how dogs experience the world. Having previously worked in shelters and working dog kennels, Mia specialises in research that translates into practical outcomes to benefit dogs and the people who work with them. Over to you, Mia.
Have you ever looked at your dog lying on the couch and wondered if they're resting and relaxed or desperately depressed? Australians love our dogs and we spend over $20 billion each year trying to make them happy. But how do we know we're actually succeeding? For decades, animal welfare science has focused on identifying suffering. That's really important because we now understand that animals like dogs are sentient. They have feelings and social connections that matter to them, just like us. We want to prevent suffering and promote well-being, But how can we do that? In fact, scientists have only recently begun to explore positive emotions in animals. We know the behaviour and biological signs to monitor if dogs are stressed, but what's the test for that whole body wiggling puppy joy we love to see? Science can measure a dog's eye movements, count their white blood cells, and diagnose hundreds of their diseases, but we can't tell you if they're actually happy. After more than 20,000 years of living with dogs, that seems like something we should have figured out. When I explored how people think about the welfare of dogs, my research showed that virtually everyone believes their own dog has the highest possible quality of life, while rating other people's dogs much lower. This suggests we might have some blind spots in how we assess our dog's experiences. Over the past decade, I've worked to advance the welfare of dogs in lots of places, from shelter and kennel facilities to detection and assistance dogs. While we've gotten good at spotting when dogs are stressed or suffering, the real challenge is that we don't know what thriving looks like. We can measure the bad stuff, but we've never defined the good. This matters because industries that rely on animals, like our working dogs, are under growing scrutiny. We've seen that greyhound racing is on the decline worldwide. When industries can't demonstrate genuine care for the animals they rely on, they lose our trust and it impacts their business. But here's where science can help our hero dogs. We can develop new ways to identify and measure well-being in dogs. My name is Mia, and with your help, I want to make Melbourne the home of happy dog science. With this funding, I'll be able to watch what dogs choose to do and monitor how their bodies respond to create the first scientifically proven happiness measures for dogs. Using a combination of behaviour and hormones in saliva, I can define new, rigorous biological fingerprints of positive emotions in dogs, giving us new insights about the dog on your couch and showing the way forward to understand well-being in all animals. Research suggests that dogs with choice and control are most likely to thrive. I want to develop those ideas into practical actions and tools. We can then improve training methods, how our dogs feel about vet visits, and create new technologies to help working dog agencies report on their dog's welfare transparently to the public. When we help dogs to live their best lives with us, we also end up with better sniffer dogs, guide dogs, guardians, co-workers, and companions who live well for longer, which supports all of us to live well too. After tens of thousands of years together, I think it's time we learn to recognise when our best friends are truly happy.
Hello, Mia. Thank you for sharing some more adorable animals with us.
The least we can do.
And for hopefully answering some of these big questions that people are asking, maybe on behalf of their pets. First question is about the changes that your research might drive. If we could routinely measure dog happiness, what changes do you think it might bring about for shelters, industry, or even the home?
Yeah, I think it would be a really great evidence base from which to inform good practice and best practice. I think it would offer our working dog industries that I've talked about the opportunity to be really transparent about how they're assuring welfare. I think at the moment, probably the welfare of animals is something that we don't necessarily economically value as much as we should. And there's, I guess, a growing field of research looking at that, valuing animal welfare from a monetary perspective. We know from industries like greyhound racing that it is really critical from an economical perspective, that an entire industry can pin on how well they can assure the welfare of their animals. Mm-hmm. So I think it offers a whole raft of opportunities to, I guess, assure the welfare of animals, but also inform how we can do it better.
Yeah. Now, lots of people have asked this question, and I'm sure you've had it before. This seems kind of obvious, but why hasn't anybody worked this out before? Why haven't we measured dog happiness before?
I know. It's I often joke it's like dogs were, because they were asleep on our couch, we forgot to science them. So I think initially science considered dogs an artificial species, so they weren't sort of considered worthy of studying. And I think also the fact that dogs are often in our homes made it really hard to study until just the last few decades, where we've had the introduction of things like online surveys and having portable technology that we can put into people's homes to capture behaviour and things like that. Canine science itself is a very young field within animal-based science, so only about 30 years old. which might surprise people. And the research that's been done during that time has been heavily directed by the funding. And for better or worse, the funding has tended to focus on how dogs help people. So how can we either understand the benefits they bring to us as companion animals? How do they make us healthier or us fitter? And how can we better utilize them? So how can we breed them to be better detectors or train them to be more efficient? And unfortunately, we kind of overlooked some of those basics of what do they actually need to lead their best life. And the funding in that area is very sparse. So no industry is funding that research because it doesn't sell products or create innovations.
I feel like some pet shops, as you said, people spend lots and lots of money every year to try to make their dogs happy. Lacey then has a follow-up question about what a practical tool for measuring dog happiness would look like. Are you imagining something that we'd have at home, that vets would use, different industries, working dog handlers?
Yeah, wouldn't it be great if it was just like a microchip scanner and you could just like buzz it by and get like a rating out of 10?
One of those things you have at the airport where you're just like, smiley face, sad face. Dogs
I
should
think
express that.
realistically, it's probably not going to be just one thing. We're probably going to need to look at, I guess I talk about this biological fingerprint of what happiness looks like, and it will probably need to be a range of measures. Within animal welfare science, we know that our best assessment happens when we're looking at things like behaviour and physiology and physical responses. So ideally, if I could pinpoint some, I guess what we call iceberg indicators, which are like these big things that predict what's happening on a bigger scale underneath that we can't necessarily see, that would be ideal. And that might look like a combination of behaviour or behavioural diversity. It might look like hormone in the saliva or evidence of a neurotransmitter or something like that. So we know we've got a lot of human biology that's the same as dog biology that we can sort of lean into a bit and understand. And I guess similar to the fact that up until about 30 years ago, human psychology focused on understanding disease and negative experiences and then decided, hang on, if we want to understand how people can live a good life, we actually need to understand thriving and look at that positive side. So I guess that's how I want to shift that angle and hopefully come up with something that's really feasible and practical to use. What that's going to look like, I don't know yet.
I've got one last really quick question, okay? Really quick one. I'm sorry, everybody. I know I'm running over time, but the whole Renault family wants to know what the signs are of a happy dog.
Fantastic. So a dog that's willing to engage with you and enjoy, willingly partake in activities with you. There's a really cool thing. You can look at the direction of their tail. So their tail, when it wags, it will probably bias more the one direction than the other. And some Italian researchers notice that when it's biased more to the right, that tends to indicate a happy emotional state. When it's going to the left more, that's suggesting they'd like space away from something or they're feeling a little bit anxious about things. So that's really cool. One thing you can do at home to help your dog be happier is let it sniff. So dogs live in a really olfactory-based smelly world, and if you can give them active time to sniff, that's probably more important than getting a lap around the block. Just give them 10 minutes of sniffy time to follow their nose.
Amazing. Thank you. But don't do that yet because we've got one picture to go. It's now time for me to welcome our final researcher, Dr. Simon Higham. Simon is a postdoctoral researcher at the School of Physics. Simon is developing a carbon fibre-based neural implant. He completed his PhD in biomedical engineering at RMIT, just down the road, and has had more than 6 years' experience in working with microelectrodes for neural interfacing, including neural recording and stimulation and electrochemical neurotransmitter sensing. Take it away, Simon.
What's the very first thing you did when you woke up this morning? Maybe you swung your legs over the side of the bed, placed your feet on the floor, and stood up. Simple, right? Walking on your own 2 feet. It's something most of us don't even think about. But for more than 20,000 Australians who've experienced a spinal cord injury, that simple act of standing, walking, and moving is something they've lost. Right now, there are no treatments that can truly bring back their natural movement. Here's what's fascinating though. In many cases, the body below and above the injury is still completely healthy. It's the communication that's been cut off, like a phone line that's been severed, stopping the brain from talking to the body. If we could somehow reconnect that signal by taking the messages in the spine above the injury site and relaying them below, we could restore the lost movement. That's the idea. The way we do it is through electricity. the language of the nervous system. This language is understood by tiny cells called neurons. These cells are responsible for all thoughts, movements, and feelings in the body, and it's these cells that we need to talk to so they can relay the messages. The challenge is we don't yet have electrodes small or strong enough to talk directly to the individual neurons in the spine. The spinal cord is a hard place to work. The areas we want to target are deep, they're delicate, and the spine is constantly moving. The electrodes we have now are either too big, so they aren't refined enough for fine movement, they're too stiff, and they slice into the tissue like a knife through butter as the body moves, or they're too soft, and they bend before they can slide into the places where they're needed. A material that can manage all of these demands is carbon fibre. These tiny microelectrodes, less than 1/5 the width of a human hair, strike the perfect balance. They're flexible enough to move naturally with the body, but they're stiff enough to be placed exactly where they need to go. They're durable, safe, and gentle on the tissue, meaning the body won't reject them. The only issue is carbon fibres themselves are hard to work with, which you would expect from something so tiny. I've spent the past 6 years working with these electrodes in the brain, creating functional implants that can safely and reliably communicate with single neurons. Now, I'm taking that knowledge and applying it to the spinal cord, a similar kind of neural tissue, just with a different mission. With this funding, I'll be able to build and test an early prototype and prove that these electrodes can work in the spine. Because imagine this, one day, someone who previously couldn't wakes up and swings their legs out of bed, stands up and walks again, all thanks to tiny carbon fibres restoring the conversation between their brain and their body.
Thank you, Simon. That's a beautiful thing to imagine indeed. Ben has a question, though, about the carbon fibre implants. They're wondering how the body would respond to them over time. You know, what would the biocompatibility look like here, do you think?
Yeah, so the main reason we use carbon fibres is because they're really small, really flexible. They're made out of carbon, so they're very chemically inert, and because your body's mostly carbon, your body's very used to it. So the main problem with a lot of other implants, the brain and the spinal cord, they're very good at knowing what's not supposed to be there. So if there's anything that's too large or too stiff, the immune system reacts to it, and then it creates a scar around the electrode, where carbon fibres are small and flexible enough that the tissue doesn't know it's there. So you're able to put these electrodes in, and they just move freely with the tissue. So it's the free movement that really means that if the immune system doesn't know they're there, they don't get rejected, and then they can just stay in there for years.
It's quite the feat to outsmart the immune system. That's amazing. Sally's wondering what other kind of movement you think could be restored using this approach, like fine motor function or balance, as well as sort of that large sort of mobility?
Well, so that's the whole point of using the carbon fibres. Mm-hmm. So right now, a lot of the electrodes that they put in are quite large. So the larger the electrodes you have, the less refined the information you're getting. So if you put in a lot of little electrodes, you're hearing a lot of the small conversations that the neurons are having. So if you think of the spinal cord, it's just connecting the communication from the brain through to everything else. So if you're trying to walk and you need to move many different muscles to be able to do that, you need to be able to control everything very small, very directly. Mm-hmm. If you just put a big electrode in, stimulate over a large area, you're going to end up moving everything because everything's going to fire.
Yeah.
And then to bring it back to the balance issue, the same electrodes that can stimulate can also record. So you're not just providing the movement, you're also getting the sensory information as well. So it's really creating a closed loop system. So then you can actually get a lot of the function back.
Yeah, I get that. Okay, interesting. Riley's got a technical question.
Yep.
Hold on. Well, maybe it's not technical. It looks technical to me. What's the latency of these electrodes?
So the latency of the electrodes would be that's less so on the electrodes themselves. It's more based on the implant that you're using. action potentials are it's a lot of information that the implant's going to process. So at this stage, I probably can't really say because it's not my area of expertise. But ideally it would be as instantaneous as possible, but that's going to be much more on the computing side. So I can't speak too well to that.
That's fine. That's fine. Okay, one really last quick question before they tell me to stop. From Mitchell, with the carbon fibre being so small, what frameworks are going to exist for post-implant monitoring? I guess, is there a chance that we'll lose it as well?
Well, so because the carbon fibres will be connected to the implant itself and you'll be recording and doing the stimulation with the electrodes. So you'd be in constant contact. You would know if the electrodes move, you wouldn't get any signal anymore. You don't have to see the electrodes themselves to actually know what they're doing. Okay. So you'd be able to use the implant to do that.
Phew, good to know. Wonderful. Thank you so much, Simon. And we are now at the end of our pitch presentations. A huge thanks to all of our pitchers for their amazing work and for sharing it with us tonight so eloquently. I'm just going to give everybody a bit of a round of applause here. Well done. Everyone in the studio is clapping as well. What this means is that voting is now open for the People's Choice Award. Please make sure you have a say and vote for the pitcher that you think deserves an extra $10,000 to help with their research. Please follow the link in the chat or in the QR code that's coming up. Let's see if I have the power now. All right, we need to give our judges a few minutes now to confer. Judges, are you conferring? Keep conferring. Okay, but I want to test, while they're doing that, how well you've been listening. I know we didn't tell you there was going to be a quiz, but there's going to be a quiz. No prizes for you, unfortunately, but pride and glory is on the line. So our online team is now going to share a quick Zoom quiz for you. Get your thinking caps on. Every question is based on tonight's pitches. So there are 7 questions you should be able to see on your screen now or very soon. Once you've answered all the questions, you'll be able to see the answer, but I'll read them out loud just to give the judges some time to really make their final decision. Okay, the first question is about Gaia's pitch. What did she compare the herpes virus to? Was it fire spreading uncontrollably, weeds in a garden, rocks blocking a river, or clouds in the sky? Well, I'm loving that return to meteorology there. See what you can remember which analogy Gaia used to think about how the herpes virus kind of hangs around in our marsupials. The second question is about Jason's pitch, and Jason mentioned what the brain uses to send messages between its cells. Was it A, blood vessels, B, electrical signals, C, muscles, or D, bones? It wasn't electrodes, a fifth the size of a human hair. That was, Simon. Think about it. What does the brain use to send its signals? The third question about Catherine's pitch. Now, Catherine's research started with a single atom. But just how small is an atom compared to something that we can see? Is it about the size of a grain of sand? Or is it 1,000,000 times smaller than a tennis ball? Is it 1,000,000,000 times smaller than a tennis ball? Or can you see one on my finger if you squint really, really hard? If you've already answered all the questions, if you've been very speedy and got the answers, no spoiling, please. The fourth question is about Kim's pitch. Now, which country did Kim not mention as a place where fog harvesting occurs? Was it A, Chile, B, South Africa, C, Canada, or D, the USA? Have a think. That's quite a tricky one. Okay, question number 5 from Megan's pitch. If you put the average sow, a mother pig, on the bathroom scales, she would weigh about as much as A, a standard fridge, B, a fully loaded washing machine and dryer stacked on top of each other, C, 3 microwaves, or D, a queen-sized bed and a mattress? You paying attention to that one? Put it in. All right, question 6 about Mia's puppies and Mia's pitch. Now, what surprise did Mia discover in her research exploring how people think about dog welfare? Was it A, dog owners think all dogs are equally happy? B, people think working dogs are happier than pets? C, virtually everyone thinks that their own dog has the highest possible welfare? Or D, people can't tell when dogs are stressed? Think about that. You can confer with your four-legged teammate if you like. And finally, Simon's question, and I realise I've given away the answer. So you have to have been paying attention to me and Simon. What is the size of a carbon fibre? Is it less than 1/5 of a human hair? Is it the size of a human hair? Is it half the size of a human hair? Or is it the size of a needle? Now, you hopefully have completed the quiz and you can see the answers on your screen. But for those of you who have not done that, and you think quizzes aren't for me, I'm just going to tell you the answer, okay? So, Gaia compared the herpes virus to weeds in a garden. You'd think you'd kill them, but then they come right back. Jason told us that the brain uses electrical signals to send messages between its cells. Answer B. Catherine's research starts with a single atom, and an atom is 1 billion times smaller than a tennis ball. That's a number that I can't even fathom. Kimberly did not mention Canada as a place where fog harvesting occurs, and you know, that's an interesting question to have about why that doesn't happen. If you put an average sow on the bathroom scales, she would weigh about the same as a fully loaded washing machine and dryer stacked on top of each other. Well done, those of you who got that one correct. Mia was really surprised to find that virtually everybody believes that their own dog is the happiest dog it can be. Everybody thinks that their own dog has the highest possible welfare. And Simon working with carbon fibres, which are, of course, less than 1/5 of the width of a human hair. So well done, everybody. That was a bit of fun. Love a bit of Tuesday night trivia, no matter where you are. I hope you went okay with that and maybe learnt a thing or two as well. How are we doing over here, judges? Have you conferred?
It's tough. Yes. I feel unwell. It's so hard.
All right, Todd and team, please tell us how was your decision-making process and who's won the top 3 of the Big Science Pitch?
All right. Thanks, Lyndon. It was really hard. Really hard. Well done to all of our finalists. 7 amazing pitches. You gave us a really tough task, and Nate is feeling sick, and I think we are all feeling a little bit unwell about this.
But also joyful.
Joyful and
also,
happy, right?
Also good.
Amazing.
Yeah.
We used a fancy rubric with 4 criteria, and these were, number 1, innovation. Was the pitch new and original? Does this idea feature new methods which are state-of-the-art or likely to make headway in the field of research? Second criteria was delivery. Was the pitcher engaging with well-explained content and persuasive arguments? Third was impact. Did the idea provide a potential significant contribution to academic achievement? Could this research support real change in understanding, methods, theory, or application? And the fourth criteria that we added in there was the question and answer session, and we really needed that to help distinguish some of these amazing pitches. So they were super difficult to rank. I wasn't given the faculty checkbook, so I couldn't give awards to everybody. They were all excellent, so we came up with a list of 3 winners, and Rebecca is going to announce our 3rd prize winner. Thank you, Rebecca.
I'm very excited to announce that 3rd prize is going to Mia Cobb.
Congratulations, Mia. Fancy check for you. Congratulations.
I'll pick up for a 2nd prize, and I would just want to echo Todd and say all your brains are astounding. Like, you're doing brilliant work, and communicating that work effectively can be a real challenge, and you've all nailed it. You've done a brilliant job. So I do hope you all walk away feeling very proud of yourselves. Our 2nd prize tonight goes to Dr. Gaya Gopakumar for her work on marsupial herpes virus. Congratulations, Gaya.
Congratulations.
All right, no, I'm good. Okay, this was really, really tricky. Genuinely, you all did such a great job. And where you fall here actually doesn't necessarily talk at all about the value of the work that you're doing. It is all so important. Full disclosure, I'm a physicist first. Two of you from the physics part of house, and of course, weather is my thing too. And piglets. But we've decided that the winner for tonight showed not only an amazing ability in communication, but also probably the broadest of potential future impact, Dr. Catherine Morgan.
Well done, Catherine.
Congratulations, Catherine. You can hold that big check there. Yay! Well done, judges. Congratulations to everybody. You can breathe a sigh of relief. Now it is time to announce the People's Choice Award. Well done, everyone who's voted. This is obviously the most exciting award. And this year, no surprises. It's been a really, really close race. I'm very happy not to be in the counting room over there. We had over 300 votes. And the winner of the 2025 People's Choice Award, again, is Dr. Catherine. Well done. There you go. 2 big checks for you. Congratulations, another huge congratulations to all of our pitchers tonight. They are all going home with $10,000 to help their research, plus our winners. I'm going to give them one last round of applause. Well done. Massive thank you to our judging panel, Todd, Imogen, Nate and Rebecca. That was a hard decision that you had to make, and I know you've put a lot of time and effort into watching the pitches and engaging meaningfully, so many thanks for that. Thanks as well to our pitchers, to Gaia, Jason, Catherine, Megan, Mia, Kim and Simon. They've done an amazing job here tonight. They've been working really hard behind the scenes to prepare their pitches, to put their stories together, to engage everybody who's watching in a really meaningful way. So we look forward to seeing what they can do next. And finally, a thank you to you. Thanks to those of you who have joined us online to support our pitches from all over the world. Thanks for asking such thoughtful questions and, of course, for deciding the People's Choice Award. A huge congratulations to everybody and thank you so much. We look forward to seeing you again at the Big Science Pitch in 2026.